Our next speaker is Dr. Ren-Ke. Ren-Ke is a professor of medicine in the Department of Surgery, Division of Cardiac Surgery at the University of Toronto. He's a senior scientist at TGHRI and works in the field of stem cell transplantation and tissue engineering. He is a Canada Research Chair in cardiac regeneration, and over 25 years his research group has defined cardiac repair, followed by cardiac regeneration to current cardiac rejuvenation. He has received several national and international awards and published over 200 peer-reviewed papers. Ren-Ke, welcome.
Thank you. Thank you, Marion. First, I'd like to thank the director for the invitation and the opportunity to share my research data with the colleagues in this TGHRI day. When you go to the conference, you always like to follow the keynote speaker because keynote speaker lay out the background. You don't need to give an introduction, it will be easy for you. But today, I guess, it's different. You're still sitting here questioning my data. Is that true or not? So I know Professor Demandis. I graduated from clinical biochemistry 25 years ago. And when I started 30 years ago in the department, he was one of the professors. And like he said, we have the journal club. He always questioned all the details we presented in the papers. So he's an excellent scientist, and he gave a very good talk. So come to my presentation. Most people in the TGHRI knows I'm working on the stem cells. I have worked on that for like 25 years. And when you're working on something for a long time, you always want to change the directions. So in today's presentation, I'm talking about conductive biomaterial. That's a material we worked last 5 years, and we feel it's a new therapeutic ways, like Dr. Demand said. But I'm going to present this data, share with you, and if you have questions, you challenge this technology, that's fine. Let me know, and I want to prove that. Okay, so I work in the cardiovascular field, and when the patient have a heart disease, and they always has a heart dysfunction. It could be multiple factors, but there's one important issue is myocardial fibrosis. When the patient had a heart failure, they could have this diffuse fibrosis between the cardiomyocytes, isolate the muscle bundles side by side. If the patient has ischemia injury, the cardiomyocytes died, and they will have a focal fibrosis in the myocardial tissue. If the patient had a severe myocardial infarction, they could have this big, messy fibrotic tissue. So the patient has a fibrosis in the heart, they could have arrhythmia. That's a major killer in the field of the cardiovascular field. When the scientists or clinicians talking about arrhythmia, they always look at the channels. For example, potassium channel, sodium channel, or calcium channel. They said these channels change, make the heart has a different action potential, has a different current propagation, therefore the patient has arrhythmia. But when the patient has infarction, when the patient has fibrosis, the major problem is this fibrotic tissue has different conductive properties as cardiomyocytes. When the patient has infarction, their normal myocardial tissue in this area, they function normal. Their action potential goes in the normal ways. But when they try to pass through this action potential to the next cells, they are isolated by the fibrotic tissue. They are not conductive. Therefore, these action potentials or current will go around of this cluster of the myocardial tissue. They will do this in re-entry. Therefore, the patient will have the arrhythmia. The cardiologists will see that, and they said, okay, what I can do is I can use this ablation to kill all these cardiomyocytes so they will not get this in re-entry. Therefore, the patient will not have arrhythmia. So as a scientist, we have to think about other ways. We know the problem is because this fibrotic tissue is not conductive. If we can find the way to change the conductivity of the fibrotic tissue, we could make this conductive potentials go through smoothly, and the cardiomyocytes will not have the re-entry, and we may diminish the arrhythmia occurrence. So that's our ideas. To do that, like 5 years ago, 6 years ago, we identified a material. It's called conductive material. So conductive material, so the conductive material, there's several conductive material. So what we focus on is a parallel. And when the parallel form together to form the polyparallel, they become conductive. So the unique of the conductive material has these structures. They have a double bond, single bond, double bond, single bond. So in this double bond, they have one pair of electron is not that stable. So when the material is doped by the oxidant, and they will remove one electron, and then they have another electron is floating along this material and make the material become conductive. So it's good they can conduct it, but when we have this material, we find it out. This material is rigid. It's very hard when you form the membrane. So you cannot apply this material into the biological system. So we have to modify this material, make it more biocompatible. So to do that, we linked this conductive material onto the biomaterial called catechin. So what we did is we use a catechin as a backbone. And the catechin has many side chains. We link this polyparallel, the conductive material, onto this set chain, and then we form this hydrogel, and this is conductive. We believe it's conductive. So we had to use a scientific way to prove that. But before we prove the conductivity, this is a different concentration of the material we synthesized. So before to prove their conductivity, we have to make sure this material is biocompatible. So one way to do that is we grow the cells on the top of material. So we coat the culture dish with a biomaterial, and then we grow on the top of the material with cells, and we can see the cells on the material grow in the normal way. There's no difference compared with control. So this data suggested the biomaterial is biocompatible. And then we want to know whether they are conductive. So we measure the conductivity of the biomaterial. We find the conductive biomaterial is more conductive than the regular material. So the material is biocompatible, material is conductive. So can we use this material in the biological system? To do the simple test, we coated this culture dish with a biomaterial, conductive biomaterial, and biomaterial. So what we did is we put 2 pieces of the skeletal muscle in the culture dish. So the culture dish has no liquid, no other things pass through the current. One muscle, we put a stimulator on the top. Another muscle, we put the receiver to measure the action potential. So we give the stimulation at one side, we receive the action potential on the other side. 2 muscles is not connected. So any current that goes through is through the conductive material. So if you look at this, you can see the control group with a very low action potential on the other side of the muscle. But when the two muscles set on the conductive material, other side receive the high action potential. So that means the current through this biological tissue goes through the conductive material and received by the other skeletal muscle. So that's the skeletal muscle. Can we use this one in the heart? So to do the in vivo study is complicated, and what we did is first, we did using the cell culture system, in vitro system. So this is a PhD student, TGHRI, and she's working in the lab. So what she did is she take the culture dish and she coated with a conductive material. And on purpose, she grow the cardiomyocytes in the very low density. If you grow the cardiomyocytes in the high density, you can see the cells linked together. They can pass a conductive velocity. They can pass a current through one cell to other. But if you culture the cells in the very low density, the cell has many space. The current pass through from one side to other side is very slow, like illustrated in this one. It takes a long time from here to here, from here to here. But in the conductive material, although the cells separate each other, but their conductive velocity go pretty quick. So we observe this phenomenon, and then we put this data in the quantitative analysis. So what we did is what we did, I mentioned earlier. So we coat the dish with a different material, and then we grow the cardiomyocytes in the culture dish, and then we identify the two areas. One area is an initiation point, and with the same distance, we identify the other area as an end point. So point 1 and point 2. We measure the conductive velocities from this side to this side. We count the frame, and then we evaluate the time. And in the control group, they take a certain time from point 1 to point 2, and in the control group is the same thing. But in the conductive material, from point 1 to point 2, it's very close. So that means the conductive material cushion help this cardiomyocytes propagation very fast from one point to another point. But people still can question us, you could see the cells in the different density. How about you separate two groups of the cells? So she treated another study. So what she did is she take the culture dish, coated with different materials, and in this dish, she put the metal ring in the dish, and then the ring separate the cells, and then she seeded the cells in the center of the ring and also outside of the ring. The two classes of the cells separated by the space, and then she used calcium imaging to measure this synchronization of the two classes of the cells. And the first group is a control. You can see the center and the peripheral beating at a different speed. And this is control. They are beating in a different speed. But in the conductive material as a cushion, you can see the cells contraction from outside and inside always together. So the curves showed here is the inside and outside cells contractions, and they are matched. But in the other two groups, they are beating at different rate. So this paper published in 2015. And then we have performed quite a bit study, quite a bit in vitro study to prove that the conductive material is conductive for the biological current. So we want to prove whether this material can be used in the heart. So like we mentioned earlier, first we created myocardial infarction. And then at 4 weeks later, 1 week later, we inject the different material into the infarct region. So one group, we inject the conductive material, other group are the control group. So we monitored the heart for 12 weeks. So this is a picture, enlarged picture. You can see the conductive material, which is colored in black, cover entire scar tissue. And then we evaluate this heart function. So the first question comes to us when we presented the data to the others. They said, if you inject the conductive material into the heart, you can make the heart into massive because of the conductive and every cells were beating synchronously, and then how this heart can function. So to answer that question, we use a telemetry to measure the heartbeat and heart function. And then we find it out. The three groups, the control group, the control biomaterial group, and the conductive biomaterial group, the heart rate is same over the 12 weeks. There's no difference. So the people may question why you inject the biomaterial into the heart did not change the conductive velocity of the heart. I believe the conductive capacity of the conductive biomaterial probably lower than the normal myocardium tissue. That's why the material injected in the normal region did not matching the normal conductive velocity. And then we look at the premature ventricular contractions. We find it out. In the conductive biomaterial group, we have less PVCs compared with the other two groups. That's functionally showed the conductive biomaterial can reduce the PVC. That's one of the signature of the arrhythmia. But I will have more data to show. The second study we did is we measure the action potential of the heart. So if you look at this is heart, this is infarct region, and we inject the biomaterial or conductive biomaterial into this infarct region. And then we use the MEA to measure the surface action potential from the normal region to the infarct region, and then we combine the data together. And I'd like to ask the help to show the movie, please. The left side. Yes, please. So this is a control group. You can see the action potential go to the border of the infarct region, and then they come back. The action potential cannot go through this infarct region. But in the other side, can you play the right side of the movie, please? Thank you. So after we inject the conductive biomaterial, and you can see this, can you play it again? Thank you, the right side. When we inject the conductive biomaterial, you can see this action potential goes through into the infarct region and goes through that infarct region. So that's the current propagation can go through the infarct region if we put the conductive biomaterial into that area. So that's talking about the current propagation. We also use this 8-lead catheter to measure the surface action potential. So what we did is, this is an 8-lead catheter. So we put 2 leads in the normal region, we put 2 leads in the border zone, and we put the 2 leads in the infarct region. Use the same catheter to measure the 3 areas action potential. So we just attach this catheter into the normal heart. And then you can see the action potential in all these 6 leads are same. The other 2 leads are used as a reference. But when we put this catheter onto the infarct heart, you can see the normal region, action potential is high. Border zone is similar. The infarct region, action potential is much lower. And then we put this material into our studied animals. We find that the control group, the normal region, high, similar, lower. Control group, high, similar, and lower. But when we look at this, with the influx region with conductive biomaterial, you can see high, similar, and similar. When we take the ratio of the influx region divided by the normal region, we got this data. We find it out. After we inject the conductive biomaterial into the influx region, that region action potential can increase. We also look at another parameter that's QRS intervals. That's parameters to show the patient prone to the arrhythmia. So with the injection of the conductive biomaterial, the QRS becomes shorter. That means we can cure the patient with a long QT. This is a QT patient. Those patients prone to the arrhythmia. So we also did the imaging. The imaging data is not that great. The reason for that is this imaging based on the calcium flushing combined with the dyes show the green fluorescence. So the normal heart, you can see the conductive propagation from bottom to the base of the heart, step by step. That's flow very well. But when we have this influx region, the influx region has no cardiomyocytes, therefore there's no calcium taking, so this imaging that's not great. But what we did is we look at the initiation time point and reach to the other end of the flushing. We calculate this and we find it out. The heart with the conductive biomaterial, the travel faster than the control group. We also use a clinical program to stimulate the heart to see whether those heart is prone to arrhythmia. So this is the program electrical stimulation studies. In the control group, they are more prone to the arrhythmia. In the heart with a conductive biomaterial, it's less prone to the arrhythmia. So this heart is less become arrhythmic because previously I showed they are shortened the QT time and QT intervals. So those are the data we showed. After we inject the conductive material into the influx region, we can change the nature of that area, increase the conductive velocities from one end to another end with this conductive material. So that improves the cardiac function. So that's the cardiac function analysis. So that's the conductive material make the heart become more synchronized. They can working together very well. So now come to the conclusion. We created a biocompatible conductive material. After put the cardiomyocytes onto the conductive material, we can make the cell become synchronized, working together. And after we put the conductive material into the heart, we can help the scar tissue become more conductive. So hopefully one day we can repair those wires, broken wires in the heart, and rebuild that, and the patient will have no arrhythmia problem. So that's the conclusion, and I like to appreciate the student and fellows working in the lab. They contribute a lot, and the conductive material we worked with is made a contribution by the Professor Sonya from Taiwan. Thank you very much. Thank you.
Mansoor?
Ren-Ke, thank you very much. There obviously is some intellectual property here and some potential clinical application here. I think one of the nice things that a day like today can achieve is if we share those types of stories. Can you just give us a brief review of how you have found this intellectual property translation and how that's going?
So the first paper, as a scientist, you always look at the publication. So when we have the data, we feel novel, we send it for the publication. And later on, Thomas Peirce find has a great potential and potential for the commercialization. So after that, we hold the publications and we start to initiate the fighting this the products. And I think this is a good idea. When you have the novel discoveries and talking with a business officer first, and to prevent the loss of your discoveries, potential application. And I think for this conductive biomaterial, although we use a catalyst working pretty well, and we have created a second generation of material, now we already have it. So we conjugated AMBA with a gelatin. Gelatin is more biocompatible, and now we are moving along in that direction. But we have two materials in hand, and we have proved the concept using the in vitro study and a small animal study. And recently, we just finished a 6 months long-term study using the small animals. And Tom's helped us a lot, and we move ahead. We just have a student from Rutman, and they coming to the group, help us to move on.
Hi there, Adam Gehring. Very different area than you are. I find this fascinating. It's very interesting. And I think you alluded to this a little bit, but I was talking about biocompatible materials. I was going to ask a question about degradability, biodegradation. How long in your 6 month study was that? One injection, multiple injections? How long does the material last?
That's a very good question. So, the biomaterial is a tricky word. Sometimes you said they are biocompatible, but actually when you put into the heart or put into the tissue, they could initiate inflammation. And then this macrophage will chew off all this material. In our study, we have injected catalyst PPY into the heart, and at 6 months later, we still can see the biomaterial over there, and the function is still better. But the bad thing is you can see the infiltration of cells around the biomaterial. The other material I just mentioned, the AMBA with gelatin, it's a very biocompatible. You put it over there, there's no problem at all, but their retaining time is much shorter, but we are working on that. Yeah. So far, with a PPY catalyst, we can maintain the material in the heart for 6 months. 6 months.
Ren-Ke, could you comment on what would be the optimal delivery method of this material to a injured human heart?
Thank you for the question. I know there's a lot of cardiologists here, so that question should ask them, what kind of format you want to have? So right now we have two formats or three formats. Formula one is we have the hydrogels. That's injectable. So if it's possible, the cardiologist can put these hydrogels into the catheter. When they do this electrical mapping in the heart, when they find this area is not conductive, they can inject the conductive biomaterial into that region. That's one way. Second way is a lot of patients has atrial fibrillation. And the atria is very thin layers of the muscle. So we are working with partners in the TGHRI. We are thinking to use this electrical spinning to create this nanofiber membrane, and then we can put that on the top of the atrium to prevent. So we will create the uniform conductive velocity platform and diminish the atrial fibrillation. We also created a conductive 3D patch. If the cardiac surgeon want to use that repair the left ventricular defect, they can use this conductive patch, and we can grow the cells into the patch and make the cells working synchronously and also working with the remaining myocardium tissue.
Thank you very much, Ren-Ke.
Thank you. Thank you.
Our next speaker is Dr. Sonya MacParland. Sonya carried out her PhD studies at Memorial University of Newfoundland, examining the pathogenesis of hepatitis C infection, and then she completed a postdoctoral fellowship with Dr. Mario Ostrovsky at the University of Toronto, where she studied the immunopathogenesis of hepatitis C virus and HIV co-infection. Dr. MacParland was recently recruited to UHN multi-organ transplant program as an assistant scientist, and she is establishing her lab to study immune microenvironment in the liver. Sonya.
Thank you very much. So as Myron mentioned, I'm a new recruit to UHN's multi-organ transplant, and the global aim of the multi-organ transplant program is to improve outcomes for solid organ transplant recipients and to prevent the need for retransplantation. So today I'll be talking about some of the work going on in my lab towards this goal. So today I'll be talking about how we're defining what immune cells are found in the human liver, and how we're using nanoparticle-based methods to target cells in the liver, including hepatic macrophages, and the role of macrophage phenotype in this targeting. And then I'll talk a little bit about how we're looking to translate some of this information by targeting cellular drivers of processes that are central to liver transplantation, including liver regeneration. So what do we know about cells of the human liver immune microenvironment? Well, surprisingly, we know very little about what's happening in the human liver, and most of the information we have about liver immunology is informed by mouse models. And we know very well that the mouse liver and the human liver are very distinct. But what we do know from studies using immunohistochemistry and flow cytometry is that the hepatic sinusoid has liver-resident B cells, T cells, NK cells, NKT cells, and also liver-resident macrophages that are known as Kupffer cells. And I'll be talking about them a lot during this talk. So from murine studies, we know what's happening immunologically during inflammation in mice. And under basal conditions, where there's constitutive low-level LPS migrating from the gut into the portal circulation, this LPS is cleared by liver-resident macrophages known as Kupffer cells. And these macrophages then secrete cytokines such as TGF-beta and interleukin-10, which creates a tolerogenic environment in the liver with CD4 and CD8 T cells that express high levels of receptors associated with exhaustion and tolerance, and they have a very high threshold for activation. However, when these macrophages encounter large amounts of LPS or activated via detection of double-stranded RNA, CpG DNA, and FC receptor engagement, these macrophages become activated, they upregulate MHC, and they begin to secrete cytokines such as TNF-alpha and IL-1 beta, which leads to the activation of T cells in the liver. And so macrophages also, while initiating inflammation in the liver, they also contribute to the progression of inflammation in the liver. And in mice in particular, there are Ly6 high monocytes, which are known to be pro-inflammatory, and Ly6 low monocytes, which are known to be patrolling and tissue repair monocytes. And upon inflammation, the macrophage phenotype in the liver becomes Ly6 high. And these macrophages secrete cytokines that are pro-inflammatory, pro-apoptotic, and lead to oxidative stress. They also secrete factors such as TGF-beta and inhibitors of matrix metalloproteinases, which cause hepatic stellate cells, which are found in the subendothelial spaces, to become activated, and they begin to secrete collagen and extracellular matrix, which drives fibrosis. The really neat thing about macrophages is that even though they initiate the inflammation and contribute to the progression, they also have a role in the resolution of inflammation. So upon clearance of antigen, phagocytosis initiates the change from Ly6 high monocytes to Ly6 low macrophages, and these macrophages then begin to secrete anti-inflammatory mediators, growth factors, and tissue remodeling cytokines such as IL-10, IL-1 receptor alpha, and growth factors and epithelial growth factors. And as well, they secrete metalloproteinases, which can lead to the destruction of the degradation of this extracellular matrix, which can lead to fibrosis resolution. So we're really interested in how liver macrophages allow for the establishment of inflammation and then lead to the resolution of inflammation. We want to find ways to promote resolution and to mitigate inflammation. So in the human liver, it's been shown that human hepatic immune cells are central to the pathogenesis of liver disease. So previously it was thought that the liver was more of a graveyard for immune cells. And what we've seen recently in various studies is that T cells and macrophages and invariant T cells are all participating and allowing for the establishment of liver disease and also the resolution of liver disease. So in general terms, the macrophages in the liver can either be pro-inflammatory or they can be pro-tissue, pro-regeneration, or pro-tissue repair. And these macrophages are characterized by different surface receptors. Certainly pro-regeneration or tissue repair macrophages express high levels of scavenger receptors, and they secrete cytokines such as IL-4, IL-10, and TGF-beta. Whereas pro-inflammatory macrophages, they show high lysosomal activity, they express markers like INOS, CD25, and secrete cytokines such as TNF-alpha, IL-12, and IL-1 beta. So one of the questions we're addressing in the lab is, what's the difference between the macrophage phenotype in health and disease? And if we can identify differences in the macrophage phenotype in health and disease, maybe we can skew those macrophages towards more of a healthy phenotype and promote resolution of disease. So we can do this in various disease models, but what I'm going to show you today is some data from tumor-associated macrophages in hepatocellular carcinoma. So in this slide, what you see is in purple, these are surface markers that indicate that these macrophages are more pro-immunoregulatory or pro-tissue repair, and they're scavenger receptors. And then in yellow, it's more these are markers that are more of an M1 phenotype or a pro-inflammatory phenotype, and these are cells that aren't expressing scavenger receptors and are expressing higher levels of CD25 and CD86. And what we see in this figure is that compared in macrophages found in tissue, liver tissue that has HCC, there's more of an immunoregulatory or pro-tissue repair phenotype. And it's known in the literature that if you have a tumor where there's more of these pro-tissue repair macrophages, there's actually a poorer prognosis for that patient. So what we want to do is just kind of uncover the differences between macrophage phenotype and different types of liver disease, and then we want to take advantage of these differences to try and promote a more healthy immunological environment to lead to the resolution of this disease. And so one of the neat ways that we can target macrophages is using nanomaterials and nanoparticles. So the very cool thing about nanoparticles is that they have a lot of design flexibility. So these nanoparticles can have a targeting ligand, they can have a tracking ligand, they can have a therapeutic agent either within the nanoparticle itself or on the surface of the nanoparticle. And in theory, they can be targeted to any site in the body, and they are preferentially taken up by tumor tissue because of the leaky vasculature and inflamed tissue because of leaky vasculature. So in theory, they're kind of a perfect treatment. However, in practicality, nanoparticles accumulate avidly in the liver. So up to 99% of nanoparticles that are injected into an animal will accumulate in the liver, which is very bad for targeting places outside the liver, but very neat for targeting liver disease. So what we wanted to do is look at how nanoparticles accumulate in the liver and in what cells that they accumulate. So we wanted to elucidate factors responsible for nanoparticle uptake in the liver, and then evaluate factors that can modulate nanoparticle uptake in the liver. And of course, look at intrahepatic distribution of nanoparticles in the liver. And this was all done in a rat model. So we looked at broad cell populations in the liver and found that Kupffer cells, or liver-resident macrophages, were particularly adept at taking up nanoparticles. And interestingly, there was something particular to liver macrophages because since liver macrophages took up significantly more nanoparticles than splenic macrophages. So, and also there were other cells that we didn't identify that took up nanoparticles. So this is kind of more of a push to identify the different immune cell populations in the liver, because there's certainly scavenger or phagocytic cells that are taking up nanoparticles. But it raised the question of what determines nanoparticle uptake by liver-resident macrophages. And the two questions that kind of came from this is whether it's blood flow dynamics or whether it's phenotype. So in terms of blood flow dynamics, it's thought that as the blood leaves the portal circulation and enters the sinusoid, it slows down. And so this may allow for more interactions between the nanoparticles and the cells. And as well, in terms of phenotype, we know that macrophages are educated by the organ in which they're found. So a macrophage in the spleen is probably not going to look like a macrophage in the liver. So we first wanted to address this by looking at blood flow dynamics. So what we did is we plated macrophages in an empty chamber and we pumped supernatant over these cells with different flow velocities. So we modified the flow velocity by 10-fold. And what we saw was that increasing the flow rate, so having more of a fast flow conditions, which might mimic what's happening in the spleen, we decreased nanoparticle uptake by 50%. And then if we slowed down the flow, which kind of mimics what's happening as the blood goes into the sinusoid, we could increase nanoparticle uptake. So it suggests that flow rate has an impact on nanoparticle uptake in the liver. We also then carried out a follow-up study in which we really looked into the polarization of macrophage and how that governs nanoparticle uptake. And what we did in vitro studies was we took macrophages that were driven towards an M0 phenotype using MCSF, and we drove them further towards tissue repair or immunoregulatory phenotype using cytokines such as IL-10, IL-4, TGF-beta. And what we saw was that as nanoparticles gained more of a tissue repair phenotype, they took up more nanoparticles. Compared to macrophages that were driven towards more of a pro-inflammatory phenotype using cytokines such as interferon gamma, IL-1 beta, and LPS. And so that suggests that you could take advantage of this difference in nanoparticle uptake to target specific macrophage populations. So this led to a model in which we showed that nanoparticles are taken up in the liver in a way that's modified by blood flow dynamics. As the blood slows in the sinusoid, nanoparticles have more chance to interact with the macrophages that are found in the sinusoid. But also, the nanoparticle uptake in the liver is also governed by phenotype. And what we see in mouse models is that periportal macrophages compared to perivenous macrophages have different phenotypes. For example, perivenous macrophages are larger, they're more phagocytic, and they express more scavenger receptors compared to central venous macrophages, in mice at least, that have more cytokines, that secrete more cytokines. So this study really brought back the question, the kind of the lingering question of really what cells are found in the human liver, what are they doing, and how do they change in a diseased liver versus a healthy liver. So one of the really cool things about multi-organ transplant is the access to really high-quality liver tissue. And as a result of a modification to a transplant procedure, we have access to tissue that would normally be discarded. It's an intact caudate lobe that has its own vasculature. So the cool thing about that is that this piece of tissue can be cannulated and can be flushed. So all the non-resident immune cells can be flushed out, and then the lobe's own vasculature can be used to perfuse that piece of tissue. So what that means is it leads to a slower, more gentle perfusion. So these highly sensitive cells like parenchymal cells like hepatocytes and cholangiocytes can be dissociated and homogenized, and you can yield viable single-cell dissociates. So using this very valuable tissue and using kind of the enthusiasm of the transplant surgeons, we're able to describe the cellular immune landscape in the liver and also the landscape of the non-parenchymal cells in the liver, so the cells that make up kind of the tissue structure. And we can do this using flow cytometry, but we can also do this using single-cell RNA sequencing. And so the very cool thing about single-cell RNA sequencing is you can take a small number of cells and look at every single transcript that's expressed in each cell. And so what happens is we take a piece of tissue from the liver, we take the caudate lobe, we dissociate it, and actually because it has its own vasculature, the dissociation is quite short. And then these single cells are introduced into an oil droplet with primer and barcode-containing beads, and there's a unique oligonucleotide sequence that's transcribed into the RNA. So every transcript from that single cell is mapped back to that original cell. Then the droplets are broken up, the library, cDNA library is prepared, and each of the cells the transcripts from each of the cells are sequenced. So this has been carried out from mouse liver, and what was seen just recently in Nature is that there are 3 populations that are discrete in the mouse liver. There's a population of hepatocytes, a population of Kupffer cells, and a population of endothelial cells. And this was quite interesting to us. And then we carried out single-cell RNA sequencing on 5 healthy human livers. And what we saw in the analysis is actually, again, the human liver and the mouse liver are very distinct. And what we saw was there was actually 15 discrete populations that came up in this analysis. And by discrete, it means that they had 10 differentially expressed genes that defines each population. And so what you see here, this is a tRNA plot, and this is just a 2D representation of complex genomic information, and the distance between the different populations indicates that they're more diverse, and if they're closer, they're more similar. And so when we looked at the literature and we looked at some gene lists that are available for immune cells, we found that there were populations, 2 distinct populations of liver-resident macrophages, and I'll show you later that we think that one of the populations is pro-inflammatory Kupffer cells and immunoregulatory Kupffer cells. We see populations of B cells, we see populations of invariant and NK T cells, conventional and non-conventional T cells, hematopoietic liver cells, and then we can see 2 populations of hepatocytes that, due to the expression of cytochrome P450 enzymes, as well as enzymes that are involved in gluconeogenesis and glycolysis, are thought to be zone 1 and zone 3 hepatocytes. And we see 3 populations of endothelial cells, as well as cholangiocytes and stellate cells. So the very cool thing about this information was that it kind of changed the way we thought about our flow cytometry panels and how we would describe, for example, liver-resident macrophage. So we previously gate on CD68 as a marker of macrophages, and this came up in both of the macrophage populations. However, the literature, and certainly we thought that CD163 was a marker of immunoregulatory or pro-regeneration macrophages. However, this came out that actually CD163 is expressed in all macrophages. It's just higher in the immunoregulatory, these macrophages that are more tissue repair. But when we looked further at the gene sets, we found distinct markers that we didn't have in our panels before, including Marco and lysozyme, that show distinct differences between the 2 macrophage populations. And this population, for example, Marco, it held true in all 5 patients. So the M2 population, regardless of, because the viability of the perfusion and the viability of the dissociation really impacts the numbers of cells you get. But for all 5 patients, Marco jumped out as the highest, one of the highest genes for this macrophage population that had more genes that encode for have immune regulatory properties. So the question is how we can translate these results. So we'll have quite a bit of data, and so what we want to do is use that data in health and then also in disease, in rejection, and ischemic reperfusion injury to define cellular targets of processes central to liver transplantation. For example, in ischemic reperfusion injury, the more severe the ischemic reperfusion injury, the more likely that graft is to fail. And also, if we can look at the role of macrophages in rejection, maybe we can create a liver pre-transplantation that's less likely to reject. And then we also want to look at regeneration. So for example, in patients that have chronic inflammation and in patients that are older, the regeneration is not as ideal or is not as fast. So we want to look at what's happening in a normal regeneration, what's happening in slower regeneration, and see if we can bring these livers closer to a healthy or a normal regeneration situation. And then if you can enhance liver regeneration, then you can actually prevent transplant by doing more aggressive resections when you have a tumor or something like that. So, for example, we're looking right now, so we have these really excellent microsurgeons in multi-organ transplant that are carrying out 70% hepatectomy. So the right median lobe and the left lobe are removed from these mice, and then we follow the mice for 7 days, and then we look at them again at 3 weeks. And we're looking at all the different immune cell populations by flow cytometry, and then we'll look at it by single-cell RNA-seq. And what we see is that in control mice, there's a nice population of CD206 positive. So CD206 is a scavenger receptor that's almost a gold standard marker, or previously a gold standard marker, for immunoregulatory macrophages. And we see that population is present in a control mouse, but not in a mouse 7 days post-hepatectomy. So we want to look at all the different immune cell changes in hepatectomy, in normal liver regeneration, and in less ideal liver regeneration, so we can have targets to shift the immune phenotype towards better regeneration. So in summary, we want to define the different cellular processes that are taking place in the liver in health and disease, and use that information, lever that information to target processes that lead to poorer transplant outcomes or that lead to the need for transplant, so we can treat and prevent the need for retransplantation. So I'd like to acknowledge all of the people in multi-organ transplant who are really super engaged in these studies and are providing liver tissue at really high quality liver tissue. Dr. Barry Gator and Dr. Jeff Liu in U of T for helping with the bioinformatics, or doing the bioinformatics work. Dr. Ostrowski in U of T immunology. All the people in Princess Margaret Genomics Center and the transplant surgical fellows, and then all the people that are working on this, Dr. Max Ma, Dr. Agata Barchak, Justin Manuel, who carry out the mouse models, all the summer students that worked, the co-op students, and then the grad students who've just joined my lab. Happy to hear any questions.
Thank you very much, Sonya. The paper's open now for questions.
Thanks. That was very interesting, Sonya. I'm curious how you would deliver nanoparticles to other organs than liver, recognizing that liver picks up most of them. Could you target any specific extracellular receptors or any antigens on other tissue to push the nanoparticles elsewhere?
Definitely. So no matter how the nanoparticle is targeted, it's non-specifically taken up by the liver. But one of the ways we can probably promote or bypass the liver is to induce a transient inflammation in the liver and downregulate those scavenger receptors. The problem is you don't want to use a gold nanoparticle, and you don't want to lead to fibrosis in the liver. But if we use something like a biodegradable nanoparticle prior to injecting a nanoparticle that's targeted to the kidney, then we could bypass that first pass effect would bypass the liver.
One of the unique features of the liver is that it's an organ that can regenerate, which one doesn't find in most other tissues. Do you know whether macrophages or Kupffer cells are required for this process, and if so, which population?
So in mouse models, there have been several mouse models that have either deleted all macrophages and they've found that regeneration occurs very poorly, or they also delete these high scavenger receptors, lots of CXCL12, those like M2 macrophages, and that slows regeneration as well. Also, if you delete things like NKT cells, it speeds regeneration as well. So it's kind of an interplay between different immune cell populations, but in the absence of macrophages, liver regeneration, and actually we've deleted macrophages using claudronate depletion, and the animals actually die after the 70% hepatectomy. So definitely.
Mina.
Beautiful work, Sonya. So given this incredible efficiency of delivery to the liver, are there any therapies in place or near place to deliver liver-specific therapies using this technology? today?
Not that I know of at the moment.
Okay, thank you very much. The last speaker in today's session is Beate Sander. Dr. Sander is a scientist at TGHRI and the director of health modeling and health economics and population health economics research at the Toronto at Theta. She's also an assistant professor at the Institute of Health Policy Management and Evaluation at the University of Toronto. Dr. Sander's areas of expertise include health economics, decision analysis and simulations, infectious disease epidemiology, and population health decision making. Dr. Sander.
All right, Sonya. Thanks, Myron, for the introduction. I'm very happy to be here today. I'm a new recruit to UHN, and given that I'm one of the new kids on the block, I thought I may give a little bit of introduction about myself, too. So who am I? I'm German. I grew up in Germany, where I trained as a nurse and worked as an ICU nurse for a couple of years before going back to school and doing an MBA. During the time, I was very interested in global health and development, and so I did a project in Papua New Guinea, which went totally unexpected, like very different to what we had ever planned, but we did get a book out of it around the traditional use of plants by the Didipa clan. So I worked with biologists there. So I went back to Germany to finish my degree and then got hooked on economics of development, and I went to Australia to do a master's degree in that, and stayed on to work at the National Center for Epidemiology and Population Health in health economics. So that's where I discovered that there is such a field as health economics, and I got really, really excited about it because, wow, my clinical training and my economics and my MBA seems to be like all coming together in one place. I went back to Germany and worked in consulting for a couple of years before making my way all across here to Canada, and that was in 2003. And my first place was actually UHN, where I started working as a research associate at the time. And later I went back to school and did my PhD at the Institute for Health Policy Management and Evaluation at the U of T. I got my first job post-PhD with Public Health Ontario, so that's where kind of my public health, population health angle comes from, and also an appointment at ISIS. And so since June, I'm back at UHN. I'm very excited to be a scientist here and work with the Toronto Health Economics and Technology Assessment Collaborative. And so in terms of research, I do economic evaluations, I do methods development, practical applications, mostly focused on infectious diseases. Current work involves evaluating Zika interventions and West Nile virus mitigation strategies using data-driven simulation modeling. So I'll be talking about that a little bit. And the other area of my research focus is using large health administrative data sets held at ISIS, Institute for Clinical Evaluative Sciences, to look at the healthcare burden of infectious diseases. Okay, so in terms of background agenda today, I will give a little bit of background. What's the context? Why do I care about economic evaluation, and why should other people care about economic evaluation? A little bit of background on the concept of population health, economics, and data-driven simulation, and then have one example around West Nile virus and finish off with some of the key points. Why do we care about economics? Because it's all about choice, right? In the healthcare system, as if in your daily life, we have to make choices, and choices are sometimes difficult. So you have to make choices in terms of public funding, fund drug A or drug B, invest in an immunization program or the new MRI. And so these decisions and choices have to be made within one category, across different interventions, across prevention, treatment, or kind of programs. And generally, you would hope that decision makers do take into account evidence. And so this evidence would include technical properties of the new technology, safety, efficacy and effectiveness, of course, economic attributes and impacts, but also social, legal, ethical, and political impacts. And depending on the intervention, context may be very, very important. And so being a health economist, I will focus on the economics today. For the non-economist, the economic evaluation really is a solution to a constraint maximization problem. So we want to maximize all the good stuff. We want to maximize health. We want to maximize numbers of patients tested. We want to improve our quality of care. But of course, there are constraints. And so those constraints include limited resources, which could be money, but could also be time, space, people. And so economic evaluation is a comparative analysis of alternative courses of action in terms of both their costs and consequences. So it's not just about the money, it's about benefits and costs. And translation would be, what's the bang for the buck, efficiency, or value? What's the value of a specific intervention? Now, moving on to population health, population health approaches really aim to improve the health of the entire population and reduce health inequities among population groups. And so in this context, we need to consider and act on a broad range of factors and conditions that have influence on our health. So from socioeconomic factors all the way to very specific healthcare interventions. And so in that sense, we will need system approaches, right? So we will need to think about integrating interventions that are within, but also that are outside the healthcare system. And so having this type of interventions, they have a lot of, I would say, specific considerations we need to take into account if we think about evaluation. So they're often not single interventions. They are often encompassing prevention treatment. That's the whole point of it. They're often non-medical and also often include large-scale policy changes. Generally address large and very diverse population groups, sometimes selected priority groups. They're aimed at risk factors, so not necessarily at a specific disease, but at a risk factor like obesity is a risk factor for many different diseases. And there's often a significant time lag between intervention and benefits. So think, for example, about HPV vaccines, where we immunize kids in grade 8. Cervical cancer is a disease that usually comes around 40, 50 years, so 30 years delay. Also, there's often a difference between who bears the risk and who benefits. Again, vaccines are a very good example for this. And it's often very difficult to identify who actually benefits from that intervention. And so all of this together makes it difficult to conduct any type of evaluations. And we can see that in economic evaluations, which are actually quite rare in this context. And the reasons for those are that these type of interventions are methodologically complex. So we need a little bit different, we need some different methods as we would apply to individual level evaluations. So the barriers are really complexity of interventions. How do we measure effectiveness, benefits, and costs? We need to talk about equity, and we need to talk about discounting. And I guess the complexity, and you may not see it very well, but so I've got population health pyramid where, on the bottom are socioeconomic factors. So those are interventions that increase population health to the greatest degree. And then as you get up, it becomes more and more individual. So I guess I made the point that population health interventions are complex, so we need system approaches which would integrate within interventions that are within and outside the healthcare system. And I want to introduce you to simulation, which is a comprehensive method for studying complex systems. And simulation would allow us to kind of have a virtual laboratory to try out all kinds of different things that are really difficult to test in the field. And simulation is not anything new, so it's been around for a very long time as a scientific tool. It's pioneered in meteorology and nuclear physics post-World War II, so it's nothing really that new. It's now indispensable in many disciplines, so it's not new across the different fields, and I highlighted some of them: economics, decision theory, medicine, epidemiology, that are particularly relevant to my work. But it's also being used in climate change, ecology, and all kinds of other disciplines. And I want to expand this in terms of talking about data-driven simulation. So data-driven really means that the simulation is determined by or dependent on a collection or analysis of data. So it's an integration of data analysis within the modeling framework. And I will talk about this a little more, so I hope it makes more sense. As an example for West Nile virus interventions, where we have funding to look at the cost effectiveness of West Nile virus intervention strategies using a computer simulation model. So the goal of this study was to, as I said, look at the cost effectiveness of West Nile virus mitigation strategies, and that's an Ontario-based study. And this is type of question where context is really, really important. So we have to choose a very specific area where we are looking at to evaluate this question. And so our specific objectives within this were to predict mosquito abundance, West Nile virus risk and transmission. West Nile virus is a mosquito-borne disease, so it's transmitted by mosquitoes. If we want to reduce or prevent West Nile virus infections in humans, we do have to think about what's going on at the mosquito level. And then estimate health economic consequences and determine the comparative effectiveness and cost effectiveness of different intervention strategies. And so the only way of looking at it that I can think of is simulation. In terms of simulation, if you ever played a computer game, some people may remember playing SimCity. I've done it a while ago, but not very recently. So this is kind of the mental picture you want to have. Except that for this type of simulations, we want to be very specific in terms of what we include into our simulation, right? So for West Nile virus, given that it's a mosquito-borne disease and the mosquitoes depend on landscape and climate and other things, we want to model different landscapes and climates. And of course, we need to include the vectors, which are mosquitoes, and hosts, which are birds and humans, and how they interact. And more scientifically, it kind of looks something like this, where we have within the blue part of the model, looking at mosquito abundance based on climate factors and landscape. So that's a statistical model looking at mosquito abundance and kind of recreating mosquito life cycles. Then we have what's the orange part, which is transmission between mosquitoes and birds and to humans. So birds are the ones that maintain the transmission cycle. And then once a human is infected, we have the green part of the model, which basically recreates the human disease history. So what happens to a person once they are infected over the rest of their lifetime? Are they symptomatic? If they are symptomatic, do they have neuroinvasive disease, or do they just have flu-like illness? If they do have neuroinvasive disease, do they have long-term sequelae or not? Do patients die because of acute disease? So those are the questions that feed into the human disease history model. And of course, we have to connect all three, because if we do want to prevent West Nile, we most likely will have many interventions at the mosquito level, right? So you could reduce mosquito abundance. We could try to minimize the contact between mosquitoes and humans. And of course, we can prevent disease in humans by vaccines, which are not available yet, but in development. And of course, we could think about how we could better treat patients that are infected and symptomatic. And so programming this type of complex model that utilizing different modeling techniques from statistical model to transmission to human disease modeling requires a large range of data. And so I've been working with public health units, for example, to understand what are the programs they are implementing, how do they roll it out, what are the costs of those programs. We've worked and obtained climate change data, climate and weather data to look at mosquito abundance, And of course, we have done a lot of work using health administrative data, and we'll talk about this a little bit more. You also reanalyze data from observational studies to fit into our analysis. And so I will talk a little bit about a sub-study that basically feeds into this, which looked at healthcare burden of West Nile virus illness in Ontario. So that's basically a costing study looking at cost of illness using ICIS data. So in terms of data sources, West Nile virus is one of those diseases that's not easily identified in health administrative data. So there are no OHIP codes, there are ICD codes, but they're very unspecific. So it's just really, really difficult to assemble a cohort of patients with West Nile virus infection. So what we did was extracting and generating a cohort of patients that had been tested for West Nile virus at the Public Health Ontario laboratories. And as you know, West Nile virus is a reportable disease, so basically most patients will be tested at PHO. And so we linked that data to health administrative data that is held at ICIS. And so that includes all healthcare resource use that's publicly funded, physician visits, emerge visits, hospitalizations, drugs, many other things. And so we used this data to estimate cost for West Nile virus patients. And so in terms of methods, in a nutshell, it's a population-based cohort study where we match uninfected patients or uninfected subjects to infected subjects using combination of propensity score and herd matching. And so what we want to achieve is we want to have two subjects that are more or less exactly the same except for having West Nile virus or not, so that we can estimate attributable healthcare costs. So really figure out which part of the healthcare costs are due to West Nile virus and not maybe to a regular checkup or treatment for diabetes or something that's totally unrelated. Looking at the longer time horizon, we want to figure out longer-term costs that go beyond, say, the first physician visit or the first hospitalization. We're using an approach that's called phase of care. And so what this approach does is really assigning the observation times of each patient into very distinct clinically relevant phases. And so patients can enter or leave our cohort at any time. And so basically from the time patient is symptomatic, at some point there will be a laboratory test because that's the basis we assembled our cohort on, right? So it's people that had been tested at a laboratory. The true index date may be a little bit earlier, so they may have had already 1 or 2 physician visits until actually a test was done. What's followed then by an acute infection or acute phase. So that's kind of when tests are done, acute disease is treated. And then there's continuing care, especially if there are patients with long-term sequelae from neuroinvasive disease, and if patients die, then there's a pre-death phase. And this maps really nicely to cost. So this is from another study which shows cost over time. And so they're generally costs following a U-shape where at the beginning we have like diagnostics, everything is going on, costs are high. Then there's this continuing phase where it's very stable and kind of checkups every now and then pre-death, costs will go up again. And so the type of findings we get is, doing this type of study, we can then plot healthcare resource use over time, for example. So in this figure, the black line are our control subjects or those who are not exposed with West Nile virus. And as you would expect, this would be stable, so stable healthcare resource use. Whereas the ones with West Nile virus, which is the red line, around their index date is where we see those really high costs. And we also see that actually towards the end, even 360 days post-index date or 1 year after, the healthcare resource use is still greater than for subjects that have not been infected with West Nile. And of course, we can break it down by the different phases. We talked about acute phase, continuing care, and death. And you can see the highest costs actually occur on the ones that die because of acute disease and mostly hospitalization costs. So this was just a brief example of one of the studies that goes into this type of simulation modeling. And that's why we call it data-driven. So I guess the key point I wanted to bring across today was that population health interventions are complex. They are difficult to evaluate because of that complexity. Economic evaluation of population health interventions are scarce and lack currently consistent quality. And data-driven simulation may be helpful in addressing these complexities. So one example was the West Nile virus study. I think it's generally a very useful approach for addressing complex interventions. So with infectious diseases for sure, where we need to capture transmission, especially in the One Health context, so be vector-borne diseases, where we work currently on other similar diseases, we work on Lyme disease, we work on Zika, also work on strep pneumo and C. difficile. So infectious diseases generally, I would say, candidate for this type of evaluations. I also want to mention two other categories where I think this is really, really helpful. So one is technologies that are disruptive in one way or another, where disruptive in a way that maybe care has to be rearranged. Maybe we move care out of the hospital into the community or the other way around, where we want to kind of try out of how that may all play out. Do we have the resources? Are there any bottlenecks? The kind of questions that we may want to answer. And the third one is basically all programs or policies that are multi-sectoral, so that are not only within the healthcare system, but where there may be either interventions that are outside the healthcare system have an impact on health, or interventions that are within the healthcare system but have an impact on all kind of other sectors, for example, education, social services, anything else. So in summary, I think there are many complex questions we need to answer, and maybe increasingly so, that do require system perspective, where I do think data-driven simulation is a great way of going about it, which also requires to engage broadly, and generally those projects need engagement across different disciplines. So thank you.
Thank you very much. We have time for one or two questions. If I may then ask, the type of analysis that you carry out, is government interested in these sorts of studies to be able to target the most effective way of delivering?
If the input is what? Sorry. Is government input?
No, is government interested in...
Interest? Oh,
yes.
Definitely. I guess, definitely, and it depends. So I do think in Ontario and in Canada, we have processes and decision-making bodies that make recommendations in terms of drug reimbursement and health technologies. So in Ontario, we have the Ontario Health Technology Advisory Committee. And there's a whole unit at the Ministry at Health Quality Ontario now, doing this type of analysis and doing modeling and trying to predict of what's actually going to happen if we would implement some technologies and use that as one input into their decision making, together with effectiveness and safety and all kind of other considerations, definitely. In terms of population health and public health, I think we're some steps behind and informing decision-making, but I would say that there for sure is quite a bit of interest, yeah. Thank you.
Okay, thank you very much. We now have the poster sessions, and after that will be lunch. So please, it'll be in the back room.
Time titular, do seu time B.